Bioinformatics.
Okay, welcome to Lecture 7. This is going to be pattern searching within genomes using the shell. Now, I just want to preface and say that this is not my content. This is content that I am essentially borrowing from Aaron Quinlan, who is a really incredible bioinformatician and is worth a follow on Twitter, or going to his GitHub page and looking at some of the other kinds of projects that he's involved with. So you are more than welcome to watch this video and pause and follow along with what I'm doing. Everything I'm showing you, you're going to have the capabilities to do. For some of the code and URLs that I'm going to be using, you can find those at the very top of the lab associated with this lecture. So when you type ls in Unix or shell or bash, whatever you want to call it, it just knows what to do, right? So if you type ls, you're going to get a list of directories. Now, I've alluded to this several times before, but these commands actually are told what to do by files that are housed in very specific directories. Kind of a typical architecture is below the root one level, there will be a bin folder where very important things are, like ls, head, grep. There will also be typically a second set of bin directory within the user directory as well. So oftentimes there will be multiple places where some of these commands are pulling from or could pull from, and they'll just pull from the first one they run into that's workable. And so you can figure out where they're actually pulling from by using the which command. So if you say which ls, it's from user bin ls. If you say which grep, it's from user bin grep, etc. Just to note, by convention, when you're pasting in shell or Unix commands plus the output, we always put the $sign or the command prompt in front of what we're actually executing. You don't execute the $sign. It's just so you can separate it from what the output looks like. So the path environmental variable, and we'll talk about what environment variables are today and a little bit later as well, is basically a stand-in for a whole mess of lists of different directories. Absolute paths to directories, so all the way from the root typically, although you can have them listed from the home as well. So here our path, if we echo, remember echo prints things, and here we're saying print the content of our path variable. If you put a $sign in front of something, you're saying it's a variable. And so the contents of this are this bin folder separated by a colon, then another bin folder, then this bow tie folder, a bunch of stuff, okay? And so that's the format of the path file, and you can put as many of these things in there as you want. Every single one of these directories is somewhere that when you type a command, your server will look for something that matches that command in one of these locations, and whether it's executable. So let's use this path as an example. If you were to execute the command cowsay and say, I am a cow, what's going to happen? I'm guessing on your computer and my computer currently, it's not going to find anything because it's going to look in this first folder. There's no cowsay in there. The second folder, no cowsay. All the way through, no cowsay. And so what it's going to do is it's going to kick back to you. And maybe you've seen this command before if you haven't installed something and you mistyped a command. Bash, cowsay, command not found. Or it might say cowsay not currently installed. You can install it by typing sudo apt-get install cowsay. Just a little tip. That very rarely ever works because that typically is only for things that are native or tend to come kind of pre-installed. Sudo apt-get install is not going to get us most bioinformatics software. But okay, so the computer is basically saying, I don't know what you're talking about. So this is kind of what we do a lot. We use Conda to install pieces of software into places and tell the computer that they're there. So when you installed Conda, Conda has its own bin file, and that's where a lot of these commands are actually being housed. So how do we, if we have commands that we want to install that are not part of Conda, how do we go about installing them? And so let's do this because I think this is a nice way of thinking about how the system actually works. So we're going to make a new command called cowsay, and we're going to make it usable on our computers. And so first thing we're going to do is we're going to go to our home directory if we're not there already. We're going to make a directory called cowbin, and we're going to move into cowbin. And then we are going to download, using curl or wget, whatever you want, this cowsay program. And then we will make it executable, as we've done before. and then we'll move back to our home directory and try to use it as a command outside of the directory, like how you can use ls anywhere. And so we'll shift over. Okay, we are signed into our server, so we will go ahead and make a directory, cowbin, cd into cowbin, and we will download cowsay. We will make it +x for executable, and then we'll cd back to our home directory. So now, if we try and execute cowsay, I am a cow. Let's try it. Cowsay, I am a cow. Still no dice. So why is that? Because it looked all the way through our path directory, and cowbin is not in our path. So if I echo path here, you'll see that we've got a bunch of stuff, but no cowbin there. So how do we get cowbin into our path permanently? What we can do is we can make it so that we, instead of having just these files as our path, we can add another one to the end. And so the easiest way to do that is whenever you want to set a new variable, you put the variable name followed by an equals sign. And so we can get kind of clever with this. There's two ways we could do this. We could copy and paste the original contents of path here, or we could say we want our path to be equal to our path variable, so all the contents of it already, with another colon, and then home/cowbin. And so if we do that, it's just going to add cowbin onto the end. So we can copy that, or we could type it. Now, this is where you can really mess things up, because if you somehow mistype your path, something like that, you can save over your path file, your path variable. And that can sometimes cause problems. It's called nuking your path. It's kind of a rite of passage that just about everybody goes through. To avoid that, though, before you do what I just did, I would echo path, and I would copy and paste the contents of this and save it into a file. You could save it with nano into your actual server, or just save it on your own local computer. So then you could always, if you forgot, you could say path equal to and paste that in and get your path back. Okay? And so notice, since I had already done it, you can see that we've got cowbin here as well. So let's go ahead and give that a shot. Cowsay, and it'll tab complete now. Cowsay, I am a cow. There you go. So this is a really useful piece of script where you can get a cow to say anything you want. So we just showed you how to set the path variable and change that. Now you always have the path variable, but you can set other what we call environmental variables whenever you want. So let's say you have a really long file name and you're going to use that file a lot. You're going to be, nanoing it or something. Who knows? What if we just made a variable that was the stand-in for that name? So here, let's just make a file. So we're going to echo some text and save it into long file name.txt. And then let's make a new variable called blah and set it equal to the path to that file. You could give it the path. You could not give it a path. If you give it a path, that means you can use it wherever. And then let's cat it. So we will echo just a bunch of text, and we will save that as long file.txt. Okay. Now we can set blah as equal to. We could do long file.txt, but that will only work in our current directory. We could go home/long file.txt. Now if we hit enter, we could cd into cowbin where we know that file is not located. And now we could cat blah. And there you go. There's the contents of it. Now, why is this helpful? Well, if you start to get a bunch of really long sequence names, like you start getting these SRR, bunch of numbers _1.fastq. Well, you could set one of those plus the path if you wanted to equal to read1, right? And so then when you're doing your trim-o-matic and you're doing single end and you want to paste in the forward read and the reverse read or something like that, you can just go like that rather than having to type the whole file name. And so this can start to make things a lot easier on you, but it can also make things more confusing. So you need to make sure that you keep track of kind of what you're doing. Something else to mention here is when you log out of your server, even if you just disconnect just your terminal from your server and sign back in, environmental variables are transient. They will disappear. So it's actually going to not be a permanent thing. So if you log out and log back in, you will not have that blah. And maybe I'll just show you. So if we exit, sign back into our server, and we want to echo blah, it's gone. Okay, so we've lost that. So they're only during your current kind of working state. All right, so next I'm going to show you a little bit more with Shell how we can start to kind of address some really basic questions and explore a chromosome from humans. And so again, if you want to follow along, these snippets of code for downloading these things are located in your lab. So here we're just going to download chromosome 22. It's zipped. We're going to save it as .gz. We're going to unzip it. And then we're going to move it as a way of just renaming it. Okay, so we will paste in that download. And then we will unzip it. Okay, and then we will move chromosome 22 to HG B37. There we go. So if we ls, we should have that, and we can head that HG, and we've got it. Okay, so first question. How could we determine how many nucleotides are in chromosome 22? So you can pause it if you want to try and figure it out on your own. But think about using some combination of grep, because that's how we can search for things, and probably some manipulation of word count. And so here, if you took the time to man grep, you would see that there were some ways of doing inverse grep searches. So if we think about the format of a FASTA file, the first line starts with a caret and then some identifier, and then everything else other than those lines are sequence data or nucleotides, right? So -v in grep will say, find me everything but lines that contain this. So here we're saying within this file, now you could also cat this file and then pipe it to grep. That's actually kind of my preferred way to do things, but I'm showing you a different way. Note that if you're going to do it this way, if you do not put these quotes around the caret, Shell is going to treat this as a redirect, and it's going to overwrite the contents of this file. So make sure you always put quotes around the carets. So we're saying within this file, find us all the lines that do not have a caret on them, and then pipe it to word count and give us the actual number of characters. Okay, so let's try that. grep-v for the caret in this HG file, and let's just without piping it first look at it. Okay, it's a lot of ends, a lot of base pairs. All right, so we'll stop that with Control-C, and what we really want is to pipe that to word count -c. And so we've got a big number here. There's around 51.8 million. Now if I were to ask you how many adenosines were there, or A's on that, so could you do that? Well, there's no reason we can't grep twice in a row. So here we're doing the first line where we get all the lines that don't include a caret. Then we're going to pass that to grep and grep for an A. Now, we don't want any line that has an A. So again, if you go through the manual of grep, which I encourage you to do, you'll see that -O says, I don't want the whole lines anymore. I just want the exact match. So just the A's that match this, basically. And then it's going to put every match on a separate line, and you can test that for yourself by dropping this last part and looking at it. But I know that that's the case. And so we can just count the number of lines that get output. And so let's try this now. We will start with our first line, and now we will also pass this output up here to grep again, and we'll grep for big A, and we only want the matches. So let's look at this output. So see, it's a bunch of A's scrolling to the bottom, millions of A's. Okay, so we'll hit Control-C and stop that and press up to go back. And what we really want is we want to count the number of lines. And so what we're seeing is there's about 4.6, 4.5 million A's being found right now. Okay, so that's cool. Okay, so let's, as biologists, take a minute and double check our work. So 42% of the human genome is GC content, while 58% is AT, and about half of that should be A alone. If we divide the 4.5 million by the total number of nucleotides that we calculated earlier, which was around 52 million, this is a typo here, it should be about that we're seeing only 8% of the total nucleotides being flagged as A. So that doesn't really mesh with what we expect from the human genome. So what's going on? Well, let's go ahead and use the -n option when we're grepping things to see if we can figure out where our A's are actually being located, and then we can look a little bit around some of those locations. So if we want to look and see where our A's are being located with that -n here, we can actually see that on this line here, 20, 10, 201, we've got a bunch of A's. So why don't we look at a couple lines around there? And so you could think about how you might look at the next 10 lines after 210.01, but I've got it right here for you. Kind of a dirty way to do it is to actually pipe to head and show lots of lines, but then say I only want to look at the last 10. So here we're looking at grepping for everything except that first caret, piping it and saying, let's look at the first 210,210 lines, but I only want to look at the last 10 of that subset. And so we really should just get those 10 lines there. And so let's go ahead and execute this. So here we've got that first line that's going to search for everything and match that doesn't have that initial caret. And then all we want to do is say we want, just the first 210,210 lines or so. So if we hit enter, we're going to get tons of stuff. But we want to say of that 210,210 lines, we only want the last 10. Okay, so if we go -10 there with tail, now we can see where that first row was we were interested in, and we can see something funny is going on here. Why are some of these capitalized and why are some of them lowercase? So before we talk about why that is, let's talk about other ways to do the same thing. If you were interested, you could grep and then use cat to get your line numbers as well. And then you could do grep for searches plus the 10 lines after it and search for that line number. Okay, so there's always different ways to do things. Catting where it prints the whole thing to the screen and adds line numbers. Then we're just grepping for that line number of interest plus the 10 lines after it. So multiple ways to do things. Okay, but what's going on with the lowercase letters? So hopefully some of you at some point in your careers have heard of Barbara McClintock. She is famous for looking at jumping genes in maize or transposable elements. Most genomes actually have them flagged as lowercase letters here. So this is where a transposable element is. Okay, so let's start to use grep with a little more flexibility, what we're starting to call pattern recognition, because until now we've been using it with just exact matching. And so grep and other regular expression uses can be far more flexible. And we're just going to start to touch on that. So we can do the same thing we did before, where we were going to look for just the results that had an A, but now we can add what's called a Boolean operator, and that's a fancy way of saying or equals to, or does not equals to. So here, the Boolean operator we're using is the pipe symbol, and it stands for or. So we want to grep for any line that has a uppercase A or a lowercase a. And then we're going to look through the results. Okay, so if we go ahead and execute this, we'll see that if we go all the way down, and if we kept going down for a long time, we would eventually see some lowercase a's. And there they are right there. So we're pulling everything out. So we could also use even more flexibility. We could match an entire sequence here. We could match lines that have this sequence or this sequence here. So whether there's this G mismatch. What we're doing here is we are putting in, just from looking at the differences between these two sequences, it's just the one base pair substitution. And so a really easy way to grep for either of these is to put the main part of the sequence, and then within brackets, this is going to be treated as a single character that is flexible. So it's going to look for an AA, TT, C, and then either a C or a G, and then an AA. And so you can try executing this with just the -O to get only those outputs, or it's sometimes nice to use the color for coloring the actual output as well. So I just want to show you what that looks like. And so here we can see where all those sequences are popping up, and that's kind of cool. All right, so this is something you could think might be useful if you wanted to search through a genome and look for sequences that you might expect to be frequent. Maybe you're trying to look for something related to EcoR1 cut sites, if that's something you're familiar with. And so EcoR1 cut sites have a very defined sequence of GAA-TTC or its reverse complement. And so we want to look for both of those. And so we could format this in two different ways. We could use grep just kind of as we normally do, and within quotes, we could put that Boolean operator in again. Now, we have to do this funky thing where we put a backslash in front of the Boolean operator to let grep know we want to treat it as a special character in this case. Whereas if we use the argument -big E to tell grep that we're going to be using some regular expression nomenclature, we actually don't have to do that. We can use that pipe just as is. So let's go ahead and try that. Okay, so here we've got that. Something to note, if you're copying and pasting from some quotes sometimes, that you will end up with quotes that don't quite look like these ones. And so it's always important to sometimes delete them and retype them if you're running into issues. So here, if we go ahead and execute that second example I had, we can see all the EcoR1 cut sites that we wanted to find. All right, so getting back to our original question, how many adenosines are there on chromosome 2? Well, we have all the information we need to answer that now. We can pull down all of the big A's and the little a's and do a word count with them. Okay, so let's try that. Okay, and then we're going to grep for our big A and our little a. And we want just the matches themselves, and we'll hit enter just to double check. Okay, that looks like that's working for us. And then we want to count the number of lines. Okay, so now we're getting around 10 million or so. Okay, so 10 million A's. Well, let's go back and double check our math. So we would expect about 24% of our content to be A's. We're getting about 20% of our content as A's. That's close, but still off by several million A's. Where are our millions of A's? And so that's because even though the human genome is something we've been working on for a long time and is considered a high-quality genome, it's still not actually totally finished. There are still plenty of what we call gaps or ends in the assembly, places where there are lots of long repeat regions where we haven't been able to get good quality sequence data. And so if you were to look and grep, and I'll let you do this one on your own, for ends, which is whenever we know there's a base pair there, like if we were doing paired end sequencing, but we never filled it in with what base it really is, that will come back as being about 25% of the genome. And so that's kind of where our missing parts are. So what if we wanted to quantify how many A's there were on each line? Now, I know this probably isn't a biology question, but this is just to show you some of the other things that we're capable of doing that eventually could be useful for biological questions. So if you wanted to quantify how many A's were on any given line, here all the A's are represented separately despite being on the same line, because that's how we've asked for it. We could use something called unique. Now, unique will take sorted data. If we have this long column here of being printed to the screen, and they all have the same identifier in the beginning of the column, and the A after that, it will collapse them all into one occurrence. So if we were to run unique on this output here, we would end up with just two lines in our output of just this line with an A and this line with an A, okay? And so here's what that output looks like if we run unique and then we look at 17 lines or so. We see that there's these lines, and some are uppercase and some are lowercase, etc. So you can see on line 205, there were both uppercase and lowercase a's. Okay. We could add the argument -c if we wanted to actually quantify how many were on each line. So if we look at line 210-205, there are 11 big A's and then 2 little a's, so it looks like there was just the start of a repeat region there. Now, the one caveat with unique is that the data have to be sorted before it'll work properly. So let me give you an example here. So if I were to make a text file, we'll just call this dna.text, and I put a bunch of letters in it. Okay, notice I have a bunch of A's in here. Now, if I were to cat this and pipe it to unique, our expectation would be all those A's would be collapsed, right? Well, we still get a number of A's. And that's because it only collapses things that are right after each other that are the same. So if we actually run that -c on here, we'll see that there were 3 A's here that got collapsed. But this A up here and this A down here were not, because there were other things in between. Other things that you should think about when you're sorting, you can get very creative about numerical and reverse sort, as we played around with a little bit during the BLAST lab. But this is all just to kind of illustrate how powerful Shell can be for kind of manipulating and just exploring large data sets. Really, the more little tricks and things like this that you start to get under your belt, the more you can do, and the more that you'll only really be limited by the types of questions you think to try to ask with the data. So for this lab, rather than having a more traditional lab, I'm going to have all this pasted in your lab as well, but on your own or in groups, and I recommend you work on this in groups, think about how and submit answers to the following 4 questions. What is the AT content of chromosome 22? So you've already figured out the A content. Okay, for today, you're going to have for your lab actually something that you'll hand in, and this will be something you can do on your own or in groups. And what I want you to do is answer these 4 questions and come up with code to do it. So this is a little different from kind of our normal way of doing things where you have all the code. So really what the first question I want you to do is figure out what's the actual AT content or AT ratio of chromosome 22. We talked about it for the whole human genome, but does it vary on just this chromosome? One tricky part here is you're going to have to figure out the ratio of AT to GC while excluding the ends on the chromosome. Next, how many lines in chromosome 22 have exactly 15 cytosines? No more, no less. What will be the code to do that? Restriction enzymes cleave DNA molecules, so this, just like that EcoR1. How many perfectly matching Hind3 sites are there on chromosome 22? Don't worry about sites that span multiple lines. Technically, those are there, but for simplicity's sake, I'm okay with you just focusing on ones that are on a single line. And then finally, how many of these cut sites are there if you think that there could potentially be a mismatch in the second position? So any type of mismatch. So if there's a little bit of wobble in that Hind3 cut site, how much does that change the number of sites across chromosome 22? Okay, so I'd like you guys to work on that, and I'll be there in lab to help you.